I'm happy to step in and talk about what the work we are doing in Frank's group, in the research group for Genomic Epidemiology. Currently doing a postdoc there, and it's actually Frank's title, but I figured a lot of the work I've been doing quite fits well under the topic of how we do global surveillance of antimicrobial resistant genes. And maybe I can also touch a little bit about what I think are the options and challenges here. Right, so a lot more recently, it's come to be recognized that we need to study antimicrobial resistance in a global context across different environments, populations, animals, and so on. But a major challenge of this is actually how do we do it, right? How do we create enough data that we can analyze antimicrobial resistance across different worlds, regions, across different environments, across multiple years even. And a lot of what Frank is doing and the research that's going on at DTU in Denmark quite fits well under this topic. So I decided today that I would talk about two major things we have been doing and that I've been involved in. And the first is how we can actually do global sampling campaigns together to collect samples and talk about, we are doing sewage surveillance with a data set that's been building over the years. A lot of quite more than 1,000 sewage samples over a period of 5 years we currently have. But also how, what I've been doing on my PhD, how we can reuse what other people are doing. So how we can utilize what's already been done, what's already been sequenced. So sewage is quite relevant these days. The pandemic really showed, right, the potential that is out there of using our wastewater to surveillance. So the Global Sewage Surveillance Consortium was established, I guess, a little bit before 2016, where the goal was to explore how we can use sewage for monitoring antimicrobial resistance. This goes back a while. The first global sewage paper came out in, I think, 2019. It described the samples that were collected in 2016. This is a pilot study, but actually from 60 different countries. And that actually grew into the second global sewage paper, where we were then working with almost 800 sewage samples from 101 countries, now collected between 2016 and 2019. And then the latest iteration of global sewage is the one I've been working on, where we are now working on 1,240 sewage samples from 350 cities in 111 countries that is now collected from 2016 up until 21. So I figured I'm going to start with what was observed in the first global sewage paper, where they sequenced, or they did mesonomic study, where they found actually that according to this data, Africa had the highest level of antimicrobial resistant genes, but we had the least samples from there. Furthermore, they observed that the Western world had very different resistant genes as compared to other parts of the world. That was again confirmed in the second global sewage study, where here I'm showing the abundance of all resistant genes found in the different sewage samples. The darker the color, the higher the abundance. And again, it's quite clear, according to this map, that there are different loads of antimicrobial resistant genes. And we can see in Africa and in Asia and also South America that there's a much darker color than the rest of the world. But what was much more interesting is that when you look at the basic diversity, we can actually see a regional separation of antimicrobial resistance. You can see that, I don't know if the pointer is on. Oh, anyway, we can see that we have East Asia separating or closer together with the purple ones, which is Middle East and North Africa, Sub-Saharan Africa, and South Asia, whereas North America and Europe are more clustered together, the samples. But, sorry, it really depends on what kind of resistance genes you're looking at. So here is the same PCA biplot, and then only looking at different classes of resistance. So it seems that there are different patterns depending on which kind of resistant genes you're looking at. Some are much more overlapping the different world regions, whereas if we're looking at beta-lactam, then the purple were much more separated. So here it definitely suggests that there is a regionality that drives the differences in resistant genes in sewage samples. But there's also another PC student that looked into the bacterial composition in sewage. So she did mesonomic genome assemblies. She recovered 1,400 max species, and she saw that, according to this study, that it was not the phyla that was linked to the region here. The outer band is colored by the phyla, the inner band is a world region, and you can see, right, it's kind of a muddy color. So it suggests that these bacteria actually spread globally, but they looked into, okay, they did a remapping of the reads back to the max, and so yes, it's confirmed it's not the region that can explain where these phyla is found. Moreover, it's actually the function. So they did look at the genes, and they actually found that the genes associated with membrane functions could more easily be explained by region than the phyla. So it suggests that these max found in sewage actually, they might be all over, but their function depends on which environment they're found in. And we were thinking of this and actually thought, okay, we see that regionality drives resistant genes, but the bacteria are shaped to the environment. And recently, there's been more focused on actually including underrepresented resistant genes, and by that I mean those genes that are maybe not become a problem yet, and maybe they are out there, they haven't been mobilized. We are typically working with those that are already been mobilized, so they've been acquired by different pathogens likely. So that's why we have the ResFinder database. It actually contains all required resistant genes that are clinically relevant, that is curated by Frank. But this is where maybe we can actually expand the picture by looking at new approaches of identifying resistant genes. So this is where functional mesonomics is relevant, where instead of just sequencing, we can actually clone in random DNA fragments into an expression host and test whether that random DNA fragment confers resistance to whatever you're screening for. And in 23 and also this year, these two papers have been published. The ResFinder FD database that contains antibiotic resistant genes that are obtained by functional mesonomics. They did a literature search and just curated this. And then there's also the paper by Deruga et al. That has studied how there might be new genes out there to antibiotics that might not yet be used a lot. So we wanted to actually compare those that are maybe already quite established and move around with those that can be identified from the environment. So we took the genes from the two papers here and combined that and did a little bit of clustering, where we're ending up working with the acquired resistant genes from the ResFinder database as one category, and then we grouped these the ResFinder FT and the Deruga genes into this group we call the FT to see what are the differences. Is there a big thing going on? And I'm just gonna say that we call the functional mesonomic genes for FT, and the acquired one from ResFinder is represented in this weird red here. So we did a screening of the 1,240 sewage samples. We can see here the abundance is plotted per country. You can see in general that there is a higher load of the FD genes across all these different countries, but it's actually quite interesting that in some places there's a big difference. For example, in Gambia, in the second global search paper, it was noted that the abundance was quite low compared to other countries in Sub-Saharan Africa. Whereas according to the FD genes, the load is actually higher. I talked a bit about already that it seems that regionality is driving the differences in the acquired resistant genes. We can still confirm this pattern. I included the PSA bipod from the second global search paper, where we can still see there's some sort of regional separation when we look at the global search 3 dataset. Picture becomes a little bit more muddy, but looking at the FD genes in the biplot, the beta diversity is, we don't see the regionality explaining the beta diversity here. We have tested it. It's actually more linked to the bacteria, the FD genes, and the acquired resistant genes. And we can also look a little bit in, okay, maybe it's not enough to look at read abundances. We can look at the sample genomes. So here I'm showing our context and what we found in them. So for the acquired resistant genes, we actually found only 452 of them. There were quite a lot of SNP variants in there, a little bit more than 6,000 SNP variants of 452 acquired genes. Whereas we saw for the FD variants, they were present in twice the amount of contexts, had a lot more variants, and like in general, we were just finding a lot more of them. But once we then look at, okay, if we compare the city sewage systems, what are they sharing? We did a UMAP clustering, and we can again see, right, that regionality is much more prevalent in the acquired variants observed in the sewage set. You see red up here, we see yellow. East Asia is grouping together. We have Europe grouping together here. But where we look at, again, at the FD variants, and I would then argue, right, this is a little bit more, the colors overlapping, it's a bit more muddy. And we're thinking, does it then make sense if we're only categorizing cities by regions? Because that is a bit of an arbitrary grouping. It's due to cultural reasons. So we figured, okay, maybe we should look at distances, like actual physical distance between two cities. So we did a distance decay analysis of here I'm showing these variants that were visualized in the UMAP clustering right before, where we have on the x-axis, we have the pairwise distance between two cities, and on y, we have the resistome similarity in what they're sharing. What I'd like to see here is that actually, yes, there is a link between distance and how similar two city resistomes are in both for acquired and FT variants. But when we look at different spatial scales, so whether the gene is just spreading around inside a country, it's just spreading inside a region, or between regions, then we can see that once the risk finder, the acquired genes, they are outside the region they have come from, the slope is just like flat. So it suggests really that once outside a world region, the acquired resistant genes, they just spread everywhere. Whereas for the FT variant, distance decay just continues to decrease in similarity. So I think there's a lot of potential in working with sewage. You can really go in and you can really trust your data quite a lot more. But I also wanted to touch about how we can actually reuse what other people are sequencing. Because when we look at how much data there is out there, there is a lot. So I like to show this graph from ENA. This is, I guess, a couple days old now, where they're showing the amount of sequencing data that is actually publicly available. This grows quite a lot. I think it has a doubling time of, I don't know, I think 33 months in this size that's out there. Currently, there's like 75.8 petabytes of data available out there, and that is a lot. And we thought back when did I start my PhD? In 2019, we maybe should take advantage of all this data that's out there. So we set out with the goal of retrieving all metagenomic data that is uploaded and publicly available. I have to say here, this graph is just every kind of sequencing data that's deposited, but we ended up downloading these 214,000 metagenomic sequencing datasets uploaded between 2010 and 2020. And to put that into perspective, this is almost 300 terabytes of data. It's 442 to the 10 to the power of 12 base pairs of sequencing reads. So it really is a huge chunk of data that is out there. We published this in PLOS Biology a couple of years ago. And one of the figures I like to highlight from this study is this one here, where we are looking at the relative proportion of reads assigned to different resistant classes. I know there's a lot of information here. We have year, location, and sampling source. But it's quite interesting that you can maybe look at European versus North American metagenomic datasets regardless of the source. See, this is kind of similar, but we can also look at oceans down here and see that we definitely don't have the same kind of resistant genes being mapped to. We can also look at, again, sources. Of course, there's a lot of human data out there, but it's quite interesting that there's also a lot of marine metagenomic samples out there, but according to this, at this point, the marine metagenomics were mainly containing finical reads. So really suggests that there is a lot to dig in here. I've done 2 published studies with this, how we can use this data. And I like to say one of them is we can really dig into it. We can look at some very few genes and really study on where they occur. We took the example of cholesterol or polymyxin resistance. I know we already heard a little bit about cholesterol. But up until 2015, it was thought that resistance to cholesterol had not been mobilized yet, but MCR1 was reported in 2015. And I think it rapidly grew in the amount of in which new genes were found. We have, I think in 2020, MCR10 was reported. I'm actually not sure whether there is an 11 yet, but I guess it's just a matter of time. But since cholesterol is a last resort antibiotic, we really wanted to see how far back we could trace these MCR genes. Unfortunately for us, we ran most of the analysis before MCR10 was reported, so I don't have it in this paper that I'm presenting here. But we're essentially asking where, when, and how much are we finding of cholesterol-resistant genes. Just to put into perspective, I said 214,000 metagenomes. Now we're looking at 4,500 of them. And we're actually quite interesting that even though that MCR9 was reported in 2019, we can actually trace it back into samples into 2007. And we can also see that the abundance has changed quite a lot over the years, and I know how many samples actually contain MCR gene fragments. We also saw that there was, again, this geographical differences in the abundance of MCR genes. Here's the total MCR gene load per country. The darker the color, the higher the abundance. It's a log scale here. And we can definitely see here that according to this data, there's quite a high load of MCR genes in Australia, also in China. I can also talk about somewhere in some places in Europe and so on. But we actually saw that once we stratified by which MCR gene we're looking at, the picture is quite different. Australia was apparently the one that had all the MCR9 genes and really driving the abundance in the Australian samples, whereas MCR1 seems to be more localized to extra Europe and China, and maybe also a little bit in South America. So this is a way to really dig into all these 214,000 samples, but it also presents an opportunity to look at everything at once. And we discussed quite a lot on how we could study it, and we came up with this idea that maybe we can study the collateral effects of resistant genes being present in different environments. By that I mean is right that the resistant genes, they can sit maybe within the same genome. They can also sit between different species, but we don't really know where they sit when we're just looking at sequencing reads obtained from genomic sequencing. So we really wanted to study, okay, what is the effect of maybe the yellow resistant genes having a higher abundance? How does that affect the abundance of the purple one? So because we saw in the big paper in PLOS Biology that maybe we can look at the bacterial diversity and the resistance diversity in all these samples, but it actually became much more interesting once we colored all these different 214,000 samples by what host the sample was obtained from, and saw that even though that, for example, I think soil sample is maybe not included here, but that in chicken sample seems there's a high diversity of resistance, even though there might not be so high diversity of bacteria. So we came up with this idea that we wanted to look at the correlation between abundances. And this is a lot of, if you go look up the paper, it's a lot of networks, but we propose that we can actually use these correlations as a way to create risk profiles or what you can have of surveillance targets. So here is this risk profile I mentioned for glycopeptide resistance. So it shows you different environments and what is the link or correlation between tetracycline resistance and glycopeptide. The more purple the color, the higher. So we can, for example, see that across different environments, there's different risk of these collateral effects occurring. One example is quite interesting is the pigs, because they've been studied quite a lot, especially by Frank, that we can look, for example, seeing the link between glycopeptide and macrolide resistance. And we actually went in and looked at what genes is it that are correlated according to analysis. The two yellow ones are the two glycopeptide resistant genes that were observed here in the pig correlation network. We saw here's the macrolide one in purple. And actually it's been shown that these genes are genetically linked, and there has been a link in the abundance, so that Frank actually showed that when you decrease glycopeptide, the macrolide will also be affected, or the other way around. So I think there's a lot of opportunities here in how you can study it. There's a lot of work going on here. I tried to keep it brief, and I also had a talk with Frank about what is his thoughts on how we can use all this kind of different data analysis approaches to analyze resistance. I talked about sewage, so talk about using everything with a focus on metagenomics. And I think one of the strengths of just working with the sewage dataset is that you are much more sure what's going on. I like to say it's the same experimental protocol, it's sequenced the same way, it's mapped the same way, and so on. It's handled everything the same way. Where if I'm just downloading what everyone is doing, I have no idea how the samples actually were handled. We can also talk about, right, we know where the samples are from. There's targeted coverage, right? We have collaborators that actually went and took the sewage samples. Whereas if we're reusing, we might get samples from all over the world, but they are from very different random environments. We had a lot of panda metagenomic samples from 1 year, for example. In the sewage dataset, you have a lot more control of the metadata. Whereas when I'm relying on what people enter when they share the data, it becomes a lot more messy. I can talk a lot on how much time I spent looking up different ways of spelling cities and coordinates and so on. Typically, when you're working with these kind of sampling effort campaigns. It's maybe it's one environment, two environments, right? We have the sewage is just one kind of environment where it really is an opportunity when you are downloading everything to study persistence across multiple environments and years and sources and so on. I would argue when you're working with 214,000 sequencing datasets, and with all the reasons listed above, there is a lot of unknown biases introduced because I have no idea of how to look into all this and reduce bias. But I hope you have less bias in your data set when you're working with this targeted sampling of sewage surveillance. I want to touch very briefly before I end on how easy it is to handle. 1,200 metagenomic samples is, of course, a lot to handle. However, it's a lot easier, and you can really go and scroll through your metadata and really figure out why it's entered incorrectly. It's easier to handle computationally, where it really requires a lot of computational resources to be able to remap and so many sequencing reads that I mentioned. But I actually think these two are coming closer and closer. When I think about it, there's a lot of opportunities. Of course, there's a lot of challenges as well. Because, for example, I know this is for a UK consortium, but wastewater has gained a lot of attention. Again, it's also been shown a long time ago. And we have, for example, the Urban Wastewater Treatment Directive from the EU that's actually mandating all member states to start collecting sewage and to monitor, among other things, and some global resistance in there. But of course, this requires more knowledge on how to handle the data. And I think this, I already talked about metagenomics. We also have whole genome sequencing and a lot of other different data sources out there. And I think it's just a matter of time before everything gets studied all at once. So there's definitely a lot of opportunities and challenges by expanding with more data sources. I think it will be very cool to see how big studies will go. Then I'm just gonna acknowledge a few people here. Of course, Frank, his main driver in a lot of these studies I've shown. Also Patrick Monk and Thomas has been my supervisors through all this, and I have a few collaborators I also like to highlight. And then here's a list of all the studies I mentioned. So thank you.